this on. Thank you, Gautam, and thank you for your invitation. Thank you for your invitation. And so you here now? Okay. I have a title of my talk like this, Establishing the Self-Sustaining Database for Sustainable Society. But as you can imagine, I'll be mostly speaking about KEC, but also I'd like to sort of convey a certain message about the database in the future. Let me start with this, with my long personal history of doing databases. As Gautam mentioned, I went to Los Alamos in 1979, and later that year, Walter Gold at Los Alamos started a nucleotide sequence database named Los Alamos Sequence Library, which later became GenBank. Before that time, I got a degree in physics department, and I was doing the modeling or explaining experimental data by a model, like it's basically a physics-based model. But from that point, I think I made a change from model-driven to data-driven approach. Around 1990s, starting around 1990s, I got involved in the Japanese Human Genome Project. And within that project in 1995, we started the KEC database, which I still continue to develop. And during this period, I started thinking about the role of database as a model of the real world. What we are doing in KEG is to represent biological information systems in the cell and organism within the computer, so that people can use this, especially as I will explain later, especially by the method of KEG mapping. But this type can be extended in a more general way. So I hope I can convey to you my message is that the database can be a model of the real world in my talk. So I'll speak three subjects. First, database as a model of the real world, and then linking genomes to society. And at the end, I'll briefly mention about sustainability. The KEC project we started, as I said, in the Human Genome Project in Japan. And at that time, maybe still, many people think that genome is the blueprint of life. So if you can decipher genome fully, then you can understand everything. But I didn't like that idea from the beginning. Many people may think that the inability of not being able to understand everything from the genome is a technical issue, but I didn't think so. It's a more fundamental issue, meaning that genome is not sufficient to construct an organism or evolution, or the biological information systems in general. So the concept of behind the KEG is this. So is the genome a blueprint of life? And I think first of all, what we inherit is not just the genome, but the entire cell. And there is a cellular continuity of the germline leading to the origin of life. So unless we uncover underlying information processing systems in the cell, and the organism and the biosphere, including how they started and evolved, we will be unable to decipher the genome. So this is my basic concept about KEGG. And KEGG has been developed as a computer model. I used to say computer representation, but I like using the word model here. KEGG has been developed as a computer model of biological information systems represented in terms of molecular networks and molecular building blocks. And we have a concept about system and perturbation. So health information categories and diseases and drugs and so forth. We have developed this category of KEGG with a concept of perturbed systems. Here, I'd like to compare with KEGG model with the AI and machine learning model. So it's, of course, the ability and usage of AI is tremendously large. But this is a computational approach. The KEGG model, the main difference is the KEGG model is that we create a model manually based on human intelligence. So we select publications and what's written there about the many biological processes. We manually represent that to KEGG pathway map, for example. And so that KEGG model has we think this is a representation or a model of the real world in terms of the molecular systems, molecular level world, biological world. Currently, KEGG consists of 16 databases that are categorized into 4 categories and color-coded by 4 colors. The first category is about the molecular networks, pathway, bright, and module. And then 2 types of molecular building blocks. One we call genetic building blocks, proteins and RNAs encoded in the genome. And then chemical building blocks from small molecules to glycans and so forth. And then the 4th category we added somewhat later in the development of KEGG. It's about perturbed molecular systems, and I will explain more in the second part of my talk. It's about diseases and drugs, and also this is mostly useful for practical applications. So this is the current status of KEGG database, but when we started, we only have 4 databases, but still we have the same similar concept of representing in terms of molecular networks and molecular building blocks. So that's the KEGG model. But there is one difference of how we make this model. The essence of KEGG model is to link molecular building blocks to molecular networks. And until around 1999, we used EC numbers for this type of linking. So we started with metabolic pathways. So we draw KEGG metabolic pathway diagrams as a sequence of EC numbers. And so if you assign EC numbers to the genes in the genome, then that means you can reconstruct the metabolic pathways. That's the way we started the metabolic reconstruction. But now we use KEGG orthology identifier to do this. because of a number of problems in the EC numbering, and also we wanted to represent not only the metabolic pathways, but also non-metabolic pathways. So anyway, so the EC numbers in the past and the KO identifiers currently are the key elements to link pathway maps and molecular building blocks. So that currently, instead of using EC numbers, we draw pathway diagrams as a connection of KO identifiers. So the meaning of KO is that, so we take public materials about, for example, metabolism or signaling, and then, but of course, those experiments are done using specific organisms. So we want to extend this knowledge to other organisms. So that's why we define functional orthologs. That's the KO. So all the pathway maps are written with KOs as network nodes. So that now once the genes in the genome are given KO identifiers, you can reconstruct metabolic pathways and signaling pathways and many other pathways. This slide is a bit another explanation of what we do. So we do what we want to do is to connect genes in the genome to gene products, how gene products are connected, forming networks in the pathway, or other molecular, other objects, including ontologies and so forth. So we consider three types of generalized protein-protein interactions, and this concept was from the beginning. We used this approach. So when you think about metabolic pathways, you often think that it's a conversion of chemical substances catalyzed by enzymes. But in our view, it's enzymes are the main players. And we want to see how enzymes are connected in the metabolic pathways. So that's the way we represent enzyme relations in the metabolic pathway. And gene expression relations, we as you may know, we don't do much about DNA signals in DNA and such things. And I don't know you like this type of statement, but genome, in our view, is like a warehouse of what's important in the it's a big, like Amazon warehouse, and you just so of course each items have signals, but what's important is how to get the right building blocks at the right time, and we only focus at that level. So this is a kind of abstraction. We ignore the details of gene expression, but just how transcription factors and transcribe to gene products and how they are related. I will be using mostly actually the metabolic pathways as examples, but this particular example shows lysine biosynthesis pathway in the past, and in 1996, and then currently. The appearance is very similar. We have boxes, that's enzymes, and circles, that's chemical compounds. But the main, of course, the main difference is that the left diagram was EC number based, and the right one is KO based. And currently, all these KEGG pathway maps are shown in our pathway browser, pathway viewer. It's a local client-side application. So in this particular case, again, this is a lysine pathway map. And on the left panel, we have a list of modules, KEGG modules. I will explain in a minute. It's a conserved sub-pathways, conserved among many organisms. And in this case, I just marked with red 2 conserved pathways that were present in the original version of the KEGG pathway map in 1996. But this one I didn't include, but additional pathways, like the main one is at the bottom of RKL pathway. So this type of white pathway map we call reference pathway map. This is manually drawn. Not only metabolic pathways, but also all other pathways we draw manually. And then we use genomic information of KO assignment and then computationally generate organism-specific pathway maps. So this green coloring means that the genes are present in the genome. At the top right, that's the E. coli pathway, that pathway, and the left bottom is the Saccharomyces pathway, and so you can see that the genes, the presence of genes, is clearly well reflected on these computationally generated organism pathway maps. So this. The way we do annotation of genomes and metagenomes, we assign KO identifiers to the genes, and by reconstructing this type of maps, we can be sure that the KO assignment is also pretty good. This is just to give you the number of pathway maps. Metabolic pathway, we have a larger number of non-metabolic pathways now, but metabolic pathway maps have our main focus are still on metabolic pathways and representing with special types of pathways. I show here global and overview and structure, but I'll explain this overview map and global map in a minute. But before doing that, I would like to explain about KEGG module and reaction module and network. These are all give detailed pictures of KEGG maps, and all these three comes on the left side panel of the pathway viewer. And this is an example of citrate cycle, TCA cycle, and the red colored part corresponds to corresponds to KEGG module M00010 and reaction module RM001 and reference network N01010. 1604. So the same segment are given 3 different identifiers, which has also different meaning of the details of this pathway diagram. So the modules are it's defined by the KOs. KOs are identified by what we call K numbers. So M00010 is a sequence of K numbers shown on this slide. And reaction module is entirely different. It doesn't use any genomic information. It's just consider the chemical transformation patterns of substrate, from substrate to product, what type of chemical transformation occurred. So we represent that type of transformation in our reaction database. And we have a concept about reaction class. It's like a KO for genes, but the reaction class is ignoring the substrate specificity, but the local transformation patterns are identical. That's what we collect as a reaction class. So as genes can this M10 module can be diversified by gene paralogous genes, for example, and then that means that with recognizing different substrates, but the reaction module can still be the same because the pattern is defined locally. I will explain this network reaction network later at the second part of my talk. But I want to also mention 2 things about this slide. One is that, as I said, it's a related the reaction module and the relationship between reaction module and reaction module. In other words, genetic network and the chemical network. But the other thing I just briefly mentioned is that the TCA cycle. It's you think it's a cycle, but in reality, it's not really a cycle. It's a collection of 2 different, at least 2, maybe 3 different segments forming a cycle. So we have a M10 and M11 module, and if you analyze our KIC database, currently containing about 9,000 cellular organisms, and this we show this coloring based on the genus level combined pathway map, meaning collecting KO assignment at the genus level. And you can see on the left, it's a Campylobacter. It's only have the left part, second part of the cycle, and the Streptococcus, for example, or only have the first part. So it's apparently some organisms still contain these fragmented portion of the cycle. Or it's not really fragmented, but it's maybe more important because this M10 module on the right is, as I said, it corresponds to the reaction module. And this is a diagram showing the how these modules are related to form a metabolic network. We want to do this type of analysis to have a picture about co-evolution of dual metabolic network, meaning metabolic network viewed as a gene network or chemical substance network. And so the RM001 is the one you saw on the first part of the TCS cycle. It's actually a chain extension reaction of the oxocarboxylic acid. So it can be used in many places. And to the right, the modification of the to oxocarboxylic acids generate different types of amino acids. So this type of architecture can be seen from this type of representation. So we have what we call overview of dual metabolic network. This is the same network as you saw in the diagram in the previous slide. But this one you can see on the Keiko website as a one type of overview network. So this overview network is drawn in a traditional way. We don't have boxes. We just have circles as compounds and lines or arrows as reactions or enzymes or genes. So we are now we just started developing a new type of overview map. And this may have some relevance to this symposium. So I show this example. This is a currently released nitrogen metabolism pathway map. Ammonia is at the circle at the end of the right arrows. So from there, on the right side is the metabolism within the body or organism. It's how the amino acids are synthesized and so forth. But on the left, more like a bacterial and other pathways. And there are 5, 6, 7 modules defined on this on the left part, and also at the bottom here, this part. And as I said, each module has we have taxonomic contents, how KOs are assigned in each map, each module. And we have an interface called module table, which indicates, so there are 6 modules on the right. And this one is at the top level of 5. Well, actually, there aren't animals in this table, but fungi, plants, fungi, protists, bacteria, and archaea, and have which modules and what fraction of those organisms have the modules. I failed to explain, but this 00531 is the one I showed here as a red line. And this is called assimilatory nitrate reductions that can be found various organisms here. And the left table is more of a detailed picture of the entire distribution, but you can change the resolution then from organism level to species, genus, and higher taxonomic groups. And this table shows these 3 modules. And for example, the last one it's an anamox, anaerobic ammonia oxidation module. And this is done by a very special bacterial species. And I think this was found like 1999 or so. It's not too old. So with this type of knowledge, we can convert this pathway map into this. And this is an overview of biochemical cycle. So how in the global biosphere and the geosphere environment, how this nitrogen are converted and by what, which organisms and which bacteria. And again, I showed this coloring of assimilatory nitrate reduction and and this part is it's a nitrogen fixation, and so we started this type of presenting since we have a fairly large number of organisms now. We started accumulating or representing a different ways of what we have already accumulated to better understand more or sort of global picture of this molecular level cycling. So this is a nitrogen cycle, but we are currently working on sulfur cycle, and then perhaps we'll do methane. But carbon cycle will be difficult because it's not really biological processes only. It's a very much complicated thing. So I don't know how we can represent, but so we limit. We have of course there's a limitation. We focus on biological processes and also what we already know at the molecular level. So, but I think this can still be useful as people have been doing for using KEGG for uncovering different types of data in their data sets. So the KEGG, of course, the KEGG pathway has been the most best utilized database, and especially for annotating genomes and metagenomes. And I just show you some examples. And this genome is already within KEGG, but we just re-annotated the same genome, same metagenome recently, and this is the result. And on the left, you see the color coding of assigned functional category of KOs. So from here, you can do the mapping, KEGG mapping. And on the right, this is just, it's not really, it's a KEGG original, it's just count the number of taxonomic groups. The best hit gene was belonging to which organism group or viruses. And for viruses, we just added recently this top right color coding. This was, sorry, no, top right, no, bottom right, this one, showing which virus family are present in this metagenome. And it's not surprising that it has cyanobacterial viruses and other viruses, protist viruses. And all these organisms are photosynthetic organisms. So we have this type of virus-host relationship, which we take from the virus-host database by Hiro Ogata in our institute. But we have a family-level relationship like this. So using the KO assignment, people would do this type of mapping against, in this case, it's a global map of metabolism. It's the largest map in KEGG, containing almost 5,000 KOs and over 400 modules. And this particular picture is a representation of which modules are completely represented in the metagenome and by color coding. Green ones are the presence of KO, and in addition, the brown coloring means that it's the presence of modules. So the modules will enable understanding of functional interpretation easier. So this was a global map for metagenome environmental sample, but this may also be used for gut microbiome. And in this particular example, we just added human genome and gut microbiome to check to see which one is unique to human. I think it's green, and then which one is unique to microbiome. That's, I think it's in red, and blue is a common shared part. So that was the first part of my talk, a database as a model of the real world. And the next subject is linking genomes to society. We have extended KEGG in two ways. So we started in 1995, and then about 2010, we added KEGG Medicus. And then around 2020, the time of the coronavirus, we added the virus more extensively. So KEGG used to be a database for any cellular organism because we as I said, we generalize experimental evidence in specific organisms to other organisms by the concept of functional orthologs. But that means we human are simply one of the 9,000 organisms. But to understand diseases and drugs, we need to know more details about human genomes and genes, especially the variations. And so we introduced a more detailed picture, variant group for the Homo sapiens. And together with that, we started developing this KEGG network database. But before explaining KEGG network, this is the basic concept behind this development. So instead of gene to disease association, we focus on network disease association. So how we call them perturbed networks can be associated diseases. And so diseases are associated with perturbed molecular networks, and then drugs are treated as perturbants to perturbed molecular networks. There are many factors that can perturb human systems, human gene variants, both germline and somatic, or pathogen, virus and other pathogens, or the gut microbiome, and carcinogens and many other environmental factors. So with this view, we develop this KEGG Medicus database, or the category of the KEGG database. There are two main objectives. One is to allow research community to utilize human genome, pathogen genome, and metagenome to obtain to develop practical applications for use in society. But mostly, currently, the drug people are using mostly, I guess. But understanding human diseases is also our target. And that's the other main, not really objective, but we started incorporating drug labels. This is actually used to drug labels in Japan, both for prescription drugs and OTC drugs in Japan. And we don't integrate into the KEG database, but we have a link to FDA drug labels. That's also link-based integrated. So at least within the Japanese society, many people are now when people search drug names on the web, so the KEG page comes very top of the list, and so people just click and see what's in it. But the problem, not the area problem, but the drug labels we don't of course develop. We just obtain from Japan Pharmaceutical Information Center. We call JAPIC, and but these drug labels are tightly integrated with the KEG database. So if after the web search, if people are interested, they can go into the KEG world. But I don't think that happens many often. But some people are using for the KEG resource for obtaining various types of information we provide. So the KEG Medicus consists of KEG network, KEG drug, and KEG disease databases. And KEG network, KEG network is a new addition, I think, in 2015 or something, around that time. And we have one network it's a one-dimensional one. So we present sequence of reactions, interactions in a one-dimensional way, and a node can be a human gene or its variant, or environmental factors, so viruses, viral proteins, or drugs. And the relationships are shown at the bottom table. So we view human gene variant and pathogens and environmental factors, and drugs are perturbants. We have now a different human gene variant that we shouldn't call perturbant, but it's more like an allelic variant. But basically, when we link disease in our concept of network disease relationships, we have perturbants and perturbant networks, and then diseases. So this type of relationship we would like to establish. To do that, we have a network variation map. Again, this is we have a number of regulatory cancer pathways and so forth, but again, I take this from a metabolic disease and from sphingolipid degradation, and some certain genes. Red marked part is a gene variant. These are all enzyme defects, and from top here it's a drug. It's an enzyme therapy drug, And so for this type of representation, oh, I should have mentioned, but this green one is a reference network. This is again shown on the left panel on the KEGG pathway viewer, and then which type for these variants we link to specific diseases. So we used to write disease pathway maps for cancers and infectious diseases and neurodegenerative diseases, but there aren't many more pathway maps we can do. So we started doing this network development for accumulating more disease data. And this particular disease was related to this green and purple colored part. And for this network, that was the one shown in green in the previous slide. And the red ones are modules. So the network was a biodegradation, but the module in this case are biosynthetic pathway, synthesizing up to lactosyl ceramide. And this compound is linked to other maps. And one example is this. So glycosphingolipid biosynthesis, lacto-neolactoseries, and this map contains glycan antigens related to blood types. And for example, this one is A type, O type, and B type. So these are not diseases, but more like a diversity of human genome. But we think since our network variation map approach can be applied to this type of variations, we have a network variation map for allelic variants. And this is for blood type carbohydrate antigen biosynthesis. And of course, we do have this glycan structures of O type, A type, and B type structures. These are type 1 structures, but anyway, there are other types of blood group antigens, and all are related to allelic variants. And so those are the data that have not been well represented in the pathway map, and we would like to see this type of picture with a different network variation map. So the KEGG Medicap's portion is we develop a network database and together with disease database, but in practice the KEGG drug is the most used database, and also we think this is it's the highest quality within the Medicus category, and I don't go into the detail, but we do our own development of annotating drug targets, drug metabolizing enzymes and transporters, and gene variants related to the drugs for pharmacogenomic biomarkers and so forth. So this sustainability, I just briefly comment on this in two aspects. It's one, as I gave the title. One is a sustainable, self-sustainable database, and this is our story of the KEGG database. And we have. I started in 1995, and 2011 we released KEGG Medicus, and currently we get around our website with 3 million unique visitors per month. But this purple's Medicus part and largely come from Japanese society. And but one interesting thing I would like to mention is this one. This is how we get the funding and how we lost the funding. And we had a very good time. This is this is of course the Japanese yen is now very cheap, but I got like $4 million per year, but now it's a very small amount. But it doesn't really affect our activity because we had a mechanism of KEG licensing, and we have currently we have in 2000 we established a Pathway Solutions. It's a private company I established, and this was a bit may not be possible like other countries because of the restriction as a professor at the academic institutions. But there were I didn't do anything wrong, but I just followed the Japanese guideline, and I asked how to do the commercial licensing to the Japanese ministry and other places, but they were not helpful because the copyright cannot be. You can't make money with copyright, and people can help for patents, but people don't help for copyrights. So I ended up starting my own company, and because of this company, we could survive even the sharp loss of the funding, and I think I'm more happy now because of the many various paperworks and so forth for writing grants and so forth. But anyway, this can be possible because of the users, and we do have academic subscription, but it's but the majority of funding comes from commercial licensing, and we appreciate those companies who care to use KEG, and that's the way we can still keep going. And but so far it was okay. It was I could establish a self-sustaining database. And this is the final slide. And establishing a database for a sustainable society. So as I said at the beginning, we view database can be a model for the real world, and especially database can be a model for human health and disease and diversity, and that's the way we do our work for developing network and the Medicus resource. And also, I don't know, since we just started, we don't know how much we can do, but we started this database as a model of biosphere. And I hope to come out some useful resource from here. And the third one is it's more like my personal interest, but I showed you the co-evolution of a metabolic network, genomic network, and chemical network. So it's how the chemistry and genomes developed in the course of the evolution of both life and the earth. So that's I guess it's a long-reaching goal, but it should be an interesting topic to be pursued in the future. Finally, I'd like to thank my collaborators. Thank you for your attention.
Thank you, Minoru. Thanks very much. We have time for questions, actually, if there are any. Yes, I come with the mic.
Thank you very much for the, beautiful talk. I have a, okay, sorry. Thank you very much for the beautiful talk. I have a question regarding, it's a great database and, you have talked about variants and diseases, network drugs. My question is like how you see the future. Like there are electronic healthcare databases, right? How can this information be integrated into EHR, electronic healthcare records, to support the translational research?
Yeah, I hope so. Our approaches that we develop, but how our resources used, I don't see much, except for my personal interest, and I don't see any limitation. And e-health, and of course, can be one target. And I hope some people, and both academics and commercial, can do that, utilizing our resource.
There was another question there.
Thank you for the talk. So your carbon cycle example, is it a question that we're doing biology and the other parts of the carbon cycle simply aren't biology? Or is it that the framework for representing the pathways and so on wouldn't be applicable? Or maybe they are, and would that tell us something about the value of frameworks that we use to exchange knowledge as working across domains?
See, what we have been doing is what's already known in a sense. We collect knowledge from the published literature, and but this is a reference. But you add additional data to this reference, then you may be able to find additional connections or components. So that's our hope of doing this biosphere thing again. So what we are doing is just what is already known, but you can use this anyway if you have this reaction and this organ enzyme, and then this can be expanded, or no, you can add non-biological data to this kind. We haven't really. I don't know if we can really make a carbon cycle map or not, but you can add non-biological data to this map, and then what kind of connections and what kind of linking would be identified. So that's the way we want to use. We wish our reference resource to be used.
Yeah, thank you for everything you've done with KEG over many years. It's much appreciated. A question about sustainability. So I'm just curious what percentage of your support comes from the commercial versus the federal. And also I noticed spikes in your funding. I assume that was around things like COVID. And so I'm wondering what these kinds of perturbations have done to your relative funding levels from commercial and federal sources.
Federal, it's less than 10%, like maybe 8 or so.
The spikes?
That was a long, sometime around 2000 and some time ago. And at that time, people talk about Millennium Project, and we got very big funding at that time.
Any other questions?